Hello everyone, my name is Katsiaryna and I'm here to present tumourevo, a reproducible open source pipeline designed for tumor evolution analysis from whole genome sequencing data. Understanding tumor evolution is crucial for decoding mechanisms of cancer progression, therapy resistance, and recurrence. Despite the availability of workflows for variant calling and quality control, no pipelines are tailored for evolutionary modeling. That is the gap we address with tumourevo. Cancer develops as a result of acquired mutations, leading to the emergence of genetically distinct subclones. Tumors are often spatially and temporally heterogeneous, so understanding the clonal architecture is the key for interpreting progression and treatment outcomes. With the advent of high-throughput sequencing, we can now study this evolutionary dynamics, but we need standardized scalable tools to make this feasible across cohorts. tumourevo is built as an external NF-core pipeline, ensuring reproducibility and scalability. It can run after somatic variant calling using outputs from tools already included in NF-core SAREC. Key functionalities include the variant and driver annotation, the quality control, followed by subclonal and signature deconvolution, and the genome interpreter to interpret the generated results. It supports the most major somatic callers like mutex, trelca, freebias, and platypus, as well as allele-specific copy number callers like ASCAT and sequenza. Here is the schematic of the pipeline. It starts with the variant calling outputs from NF-core SAREC, which are then passed through a series of modules. The quality control is handled by TINC and CineQC, ensuring sample purity and checking for miscode cinease. Driver mutations and variants are notated via tools like WEP, and subclonal deconvolution is performed using tools like PyClone, Viber, and Mobster, followed by clonal tree reconstruction using the C3 tool. And the signature deconvolution step then extracts the mutational processes using tools like SIG-Profiler and sparse signatures. So this modular design ensures flexibility while still providing a standardized framework. So we tested the pipeline on a colorectal cancer cohort with 2 patients and 2 samples for each patient, and total computational time was around 3 hours. And the quality control results show the clone-specific cinease and variant allele frequencies across samples. Here, the subclonal deconvolution identifies clusters of mutations and helps distinguish between single and multivariate evolutionary models. And finally, signature deconvolution reveals the contribution of the different mutational processes. That is the key for understanding tumor etiology. So our gathered dataset is small but enough to demonstrate the pipeline feasibility. And the next steps is to finalize the genome interpreter model to better interpret the analysis results and test the pipeline on larger and simulated datasets and improve integration with more tools and formats. So in summary, NF-core tumourevo fills a critical gap in cancer genomics workflow by standardizing tumor evolution analysis. So thank you for your listening, and I'm happy to take any questions.